Pretty eyes, pretty thighs, shawty, she a dime. Demon girl, evil eyes, she be telling lies. Slim waist.
All right, so today it is my pleasure to interview Dr. Alexander Marson, co-founder of uh, Arsenal Biosciences. So, Dr. Marson, thank you for being part of this episode. As a spearheader in your field, can we start with you taking us back and telling us about your background?
Sure. Let's see. I don't know where to start. I grew up on the East Coast. Um, I went to college at Harvard and studied neurobiology and was really interested in the intersection between science and philosophy. Worked in some amazing neuroscience labs. One of my first serious lab experiences was in Eric Kandel's lab at Columbia and got really interested in questions that I didn't expect to be. I thought I'd be studying questions of consciousness and memory and learning, and all those were interesting to me, and I was, but I got really interested in even maybe just simpler biology questions of how do different neurons in the brain turn on different sets of genes. And that question seemed tractable to me and continues to fascinate me. And that's a question that I've followed through. I wasn't sure if I was going to do research or see patients. I ended up going to medical school. Um, and then along the way, switched into the MD PhD program. And finally, I found myself in two labs at MIT, Rick Young and Rudy Yanish, two amazing labs at an amazing moment. I was studying transcription factors and gene regulation just as iPS cells were discovered by Shinya Yamanaka and got really interested in how do transcription factors work together to control cell states, cell identities, both in embryonic cells and embryonic fate choices, but also starting an interest in the immune system. And that path has now really led to what is now the current research in my lab, which is understanding how do genetic sequences throughout the DNA encoded in our immune cells control their behavior? How do they control which genes get turned on and off? But also what the cells will do. Where do immune cells traffic? How strong will the responses be? What's the nature of the response? How long will they last? And I'm interested in genetic control over all these parts of how immune cells function not just as a fundamental question, which continues to be fascinating to me, but also as a roadmap for how we can make better immunotherapies for many human diseases. How do we program immune cells to go to tumors and hopefully clear them? And how do we program immune cells to go to sites of inflammation and dampen inflammation that's causing disease or clear infection? So I'm really interested in what can we program immune cells to do to broadly treat human diseases.
Yeah, so I know you mentioned you're a MD, PhD. So, how does that shape the questions you ask in the lab and your goals? I'm just wondering because no, because I've gone a bunch of scientists here with only a PhD.
Yeah, I would say in my case, it has shaped my goals, my research questions for sure. Not so much as a plan, but just as the nature of having spent a lot of time seeing patients. I don't see patients anymore,
Uh,
but having spent years in medical school and then residency, I did a clinical fellowship in infectious diseases. And I think all of those things created an imprint of how medicine is actually delivered, what it means to take care of someone, and a different relationship with disease than I would have had if I were just reading about it in textbooks. And there are amazing PhDs who do research that's even more clinical than what I do. But for me, I've watched the arc of how my research has been drawn into questions that are not only fundamental science questions, but also have translational significance through having spent more time with patients. So I think it has left an imprint. I think the time I spent with patients in medical school also gives me a better informed position to weigh in on choosing research projects and what their potential translational implications may or may not be.
Yeah, so I just spoke to one MD, PhD person from Stanford. He was a professor, but he also said like treating patients and doing research was like it's kind of hard because you're like pretty busy. So I was wondering, is the reason why you don't see patients anymore? Is it because you didn't have the time to see patients much? Was that the reason, or was it something else?
Yeah, I think in some ways that was the core of it. I didn't have the time to do both at the level that I wanted to do it. And I didn't want to cut corners as being a doctor. And I over time watched that my heart was really in my lab, that that's where my what I was thinking about in my free time. I was thinking about the science. I was also thinking about the scientists, and I didn't want to be seeing patients if I wasn't spending adequate time thinking about their care. And so I thought, well, I should really narrow down. There was also another aspect. I went into infectious disease really focused on, well, this is what the immune system has heavily evolved to do to protect us from pathogens. And I continue to think that infectious disease is an incredibly important area of science, incredibly important area of medicine. But so much of our research has been pulled in other directions, thinking about autoimmune disease and cancer immunotherapy in ways that I didn't fully grasp when I was choosing clinical specialties. And so I didn't see, I think some people have a great synergy in their career where they're researching a disease and then going and seeing patients with it. They're learning more about the disease that allows them to help their patients better. And then as they see patients, they bring new questions back to the lab. I have friends and colleagues who do that kind of thing, and I think it's an incredible way to do science. It wasn't the experience that I was having. I felt that too often I was trying to do two things that are both hard and that were a bit disconnected and non-overlapping. So I decided to, at some point I realized that the thing I really love is the lab and the scientific team, the scientific questions, and an addiction. I actually found myself getting somewhat addicted to even small discoveries, the ability to like figure something out that you were the first one to see or to understand. And I figured that was the thing I would really double down on to try to do as much as I could in my career.
Mhm. Yeah, so I read some articles that you wrote and some other articles. So you often talk about DNA as like a programming language and engineer cells as like living medicine. So can you unpack that type of, I guess, comparison to the audience?
Yeah, so this goes back to the way I framed my path to this field in some ways. I got really interested in college in this idea that every cell in our body is more or less has the same genetic information, and that's extremes of skin versus brain versus liver have the same DNA, but they do such wildly different things. And they all have the same DNA instruction, but they enact it differently to carry out the programs that each cell has to do. Um, One of the things that drew me to the immune system was that even within the immune system, these very closely related cells, they will enact quite different programs that can have opposite effects on disease. Closely related T cells, one can be driving inflammation. You can also have the closely related population of regulatory T cells that are suppressing inflammation. And all that's encoded in our DNA and then also in the epigenetic programs that will determine which genes get turned on or off. So I continue to think of this. I think of it as there is a source code inside of our cells that is our DNA, and that code has to have a broad range of all the things that it could enact, but also has to give the instructions for what should a particular cell type do. And so that means that if we could go inside the DNA, inside an individual immune cell, you could start thinking about altering its source code, and you can actually observe the consequences. So you can actually go into the DNA now with CRISPR in ways that were not imaginable to me during my own graduate training, but now, thanks to our work and others, many others, you can actually go into that DNA and change it, and then observe the consequences of how a cell behaves differently. So you can change the code and see how a cell has a different set of behaviors. And I think this is a transformational research technology. It means that all of a sudden we don't have to look for correlations of, does this DNA associate statistically with some kind of disease trait, or does it statistically associate with some behavior? Those things are still interesting to observe in humans, but now we can experimentally often do this. We can actually change a piece of DNA and observe what are the consequences to cell behavior and and then what are the follow-on effects to those altered cell behaviors. And that continues to shape the kind of research that we do of trying to understand that code. And then we're in this era, one of the things that we've done is figure out how we can very rapidly and precisely start rewriting DNA sequences. And that means you can actually put in DNA sequences that don't even exist in nature and start programming cells to do something that they haven't beyond what they're naturally evolved to do. So immune cells are naturally evolved to survey our body for signs of infection. We can add in new code to make them survey not only for infection, but specifically program them to survey for cancer. We can program their behavior once they engage a cancer cell. So we can be more and more sophisticated in what we talk about as synthetic biology of, making an immune cell, making a cell do something beyond what it would naturally do that would be in line with a how we could treat a disease.
Mhm. Yeah, so speaking of programming, I know you met Sean Parker on Wired. There's a video of you. So I just want to ask. So I kind of saw the Mark Zuckerberg movie, and then they portray Sean away. So like, what was it like meeting Sean? Because I know people say Sean has like a complex personality or something.
Sean is an amazing person, and Sean has a very special brain. He's really an autodidact who's taught himself a broad amount, and you can just see that he's intensely curious and really smart. And he brought that in the tech world, but to his credit, I think, I mean, it's pretty remarkable. At some point, he really made a hard pivot and decided that he was also going to teach himself immunology and cancer immunotherapy development. And I would say not only has he taught himself, but he's left a pretty indelible mark over the past 10 years or so on the whole field by creating this Parker Institute for Cancer Immunotherapy that I'm a member of, that has had a huge effect on our research and played a big role in company creation and bringing things forward to I hope a lot of patients. So he has really created a whole new community and a really whole new way of pursuing cancer immunotherapies, highly collaborative across many institutions around the country. People are working together, and um, we get together every 6 months, and people show data that's not published and keep each other updated. And it's led to so many collaborations, training the next generation, and a number of companies. So Sean and I worked together really closely in the early days of Arsenal Bio. The Parker Institute, which is a nonprofit research institute, actually invested a lot of money to create Arsenal. So there's a real partnership of nonprofit creating a company, and if Arsenal does well, some of those proceeds would flow back to fund future research. So I credit Sean enormously with just a very creative, forward-looking brain about and has opened up whole new ways of doing research in biology.
So like with Sean Parker being involved in biology, do you think more Silicon Valley figures are going to be involved?
I think we're seeing that. I mean, that's
Mhm.
it's, it's so interesting. I there was a moment. I grew up in New York. I did almost all of my training in Boston between Harvard and MIT. And kind of, I think my default was that I thought I would stay on the East Coast, maybe likely stay in Boston, which has its own incredible science community. There was a moment when I came out here to start interviewing for jobs, and I just had this kind of half-baked intuition that San Francisco was where I wanted to do research. And some of it was that when I talked to scientists, I heard that there was a different way that people talked about risk. People were very willing to take risk for things that they thought were scientifically important and if they worked would be transformative. And I was really drawn to that kind of boldness. And now when I look back on it, I think a lot of that boldness actually comes from cultural crosstalk between the biology world, the science world in the Bay Area and Silicon Valley. And that kind of boldness that has led to entrepreneurship and technology bleeds through a bit to the culture of technology, of risk taking and forward-looking thinking that I think is distinctive about Bay Area biology too. And in ways that I didn't anticipate, I think that that has become not just a undercurrent, but has actually become really concrete through a number of major Silicon Valley figures who have invested heavily in biology. And so we are the beneficiaries of I mentioned Sean Parker, but also others, and notably Chan Zuckerberg, Mark Zuckerberg, and his wife Priscilla Chan have put a lot of money into biomedical sciences. And so I do think that there's some crosstalk of people who have been incredibly successful in one area of innovation, being drawn into a really different, but no less exciting and maybe more exciting field of innovation that's happening in biomedical sciences.
Is there like a cultural difference, like between like Boston VCs who are involved in biotech and like kind of West Coast Silicon Valley kind of VCs? Is there like a cultural difference in terms of investing?
I don't know if I know enough to, oh, I always think that there's probably more similarities than differences, but there might be some trends towards differences. I think what I've seen is that for years there was a dominant culture in the sort of East Coast, Boston-based, especially VCs, where the culture was heavily for academic scientists to have an idea, to be sort of academic about it, appropriately for the way that they've organized their lives, and then for VCs to come in and essentially do company creation, to come in and say, okay, like you stay in your lab, we will take this out and we will essentially own it. You'll become an advisor, but we'll take this idea and make it into a company. And I think that one of the areas where there's been an influence, I believe, from the tech world is that the San Francisco VCs have pushed back on that a bit and have really said, well, maybe our biotech founders should also be more like founders, and we should keep them more involved, have more ideas. And look, I think there's some potential for that has to be managed with their now double roles, my double role as continuing an academic lab, but also starting companies. But I do think it allows founders to play important, potentially really major roles in setting companies on the right direction early. Academics still are not largely, although there have been some notable people who've gone full-time into industry, but academics who stay in academia they're still running their lab. They're not operating inside of the companies that they found. But there are early days of companies where academics can play a big role in setting technology on the right direction, setting cultural expectations around the types of science that are done. And I think that that has filtered largely outwards from the San Francisco investment ethos.
Mm. Yeah, so yeah, I'm talking about your research. So I know your 2018 Nature paper on non-viral T cell gene editing was like a landmark. So can you talk more about it, like especially at the whole of viral vector problem that your lab was trying to solve?
Yeah. So, this unfolded. When I started my lab in San Francisco in 2013, and I knew I was interested in understanding genetic control of T cells. I didn't really know much beyond that. I wasn't really sure of what my lab would do in it, and it could not have been a better moment. In 2012, we saw the first real major successes of CAR T cells, notably with Emily Whitehead, who was the first pediatric patient treated with CAR T cells in 2012 at the University of Pennsylvania. And CRISPR technology came out, the first major CRISPR DNA editing paper from Emmanuel Charpentier and Jennifer Doudna. And I got fascinated as I was looking for around what my lab doing T cell genetics would do. I thought, well, let's put these together. This now we can actually understand genetics, but maybe we can also make genetic modifications. Not that I knew all the things that would unfold in the coming decade, but I had some sense that if you could put these things together, it'd be incredibly powerful. And we started actually the first postdoc in my lab was her name was Catherine Schumann, and she teamed up. I helped arrange this incredible collaboration with Jennifer Doudna's lab, and we early on the field and our lab included had a lot of challenge of just how do you get CRISPR into T cells. It was working well in immortalized cell lines, cancerous cell lines, and that grow well in the lab. It was growing well and it was working well in animal models. It wasn't working in primary T cells, the cells that you take out of a blood sample from a human, where we wanted it to work. And Catherine and a postdoc in Jennifer's lab, Stephen Lin, teamed up, and they very quickly hit on this solution where we could start delivering CRISPR as a protein RNA complex that was preformed, delivering that into T cells. And we started seeing early on that we could get rid of genes, and then we could also even make small changes in DNA. At that point, we could make maybe a couple of changes, maybe up to a couple of dozen DNA changes. So that set the stage for this amazing MD PhD student, Theo Roth, who came into my lab. And Theo and I agreed that his project would be, well, what is the limit? Maybe we're not limited to just a few changes. Could we just take a piece of DNA and this CRISPR complex, use the CRISPR complex to make a cut at a targeted site in the DNA, and then have a naked piece of DNA that's around and trick the cells into repairing that cut by incorporating sequences off that naked piece of DNA. There was a lot of stuff that was we and others had seen that DNA could be toxic to cells, especially T cells. Theo took this really systematic approach where he tested hundreds, maybe thousands of different conditions to get the right dosage, the right delivery conditions, the right relative concentration of DNA and protein and RNA. Finally optimize this. And what we saw was that we weren't limited to just a few changes. We could actually start putting in over 1,000 different changes. And I think from technological advances from us and others now in, this can probably be up to over 10,000 changes that can be done at this point, 7 years later. This meant that we could make CAR T cells in ways that had never been made before. The early success that was seen, for example, in Emily Whitehead, the way those CAR T cells were made was actually by taking the CAR, the chimeric antigen receptor, this artificial receptor that directed T cells against a cancer target. It put the gene for that CAR in using virus. Virus is laborious to make, it's expensive, but it's also not precise. It doesn't direct that CAR gene to a particular place in the genome. All of a sudden, we had found something that was potentially much easier to deploy, didn't require making any virus, and had the benefit of being precise. So we could introduce sequences for CARs or other types of receptors, other types of genetic enhancements, all in one piece of DNA, put it without any virus step and we could put it into a defined site in the genome. And, this is now the basis for how Arsenal cells are made. Arsenal's made a number of improvements inside the company to this. It's other clinical trials are underway at various stages of development. And so it's been exciting to watch how practical this breakthrough has been. My hope is that moving away from virus brings down the cost and also allows us to test many more designs of how to make CAR T cells rapidly to try to accelerate the process of, I think, if there's a little bit of like a technology analogy that in this idea of like the cell being something that gets programmed by the DNA. the other thing is like you can almost think of it as the DNA design is like a piece of software, and you want to be able to make versions of this. You want to make version one, version two, and be able to update this software through the lessons of seeing what works, how to make things more effective, how to make things safer. And so I hope that getting away from the reliance on virus while also adding this specificity allows us to have improved range of sequences that we can program, but also to test things more rapidly to get them better and better for patients.
Yeah, so I've heard T cell therapy, like the whole manufacturing process is really expensive. So, do you know if there are ways to like decrease the cost of manufacturing? So like maybe more companies could kind of adopt, or especially farmers could adopt T cell therapies.
Yeah, this is something that's talked about a lot. I think there's a number of avenues that are available, and I think different companies and we and others are taking different strategies that are not necessarily mutually exclusive. One is the manufacturing can just come down and be through things like automation, moving away from virus. Maybe fewer cells are required. Maybe they don't have to be grown up outside of the body and as long as they have in the past. Maybe some of the testing that's required for regulatory purposes to release these cells can be more automated. So all these things would be ways, and the extreme of this might be like almost like could you have, and people have talked about this, could you have a device that sits at a patient's bedside and a small amount of the cells from the patient's blood get zapped right there and immediately is recirculated back into the patient. And so that would be a way that, okay, you're genetically editing their cells technically outside of the body, but it's happening in a way that's very easy to implement and can be done in a scalable way. So that's one, is just to improve the manufacturing. Two is something that there's a number of companies and other trials that have been tried, has its own challenges, but is a nice solution, is what's called allo allo cell therapies. Now, what this means is allogeneic, which means that the cell doesn't come from the patient that is going to get infused, right? The opposite is autologous. So we talked about is these expensive cell therapies are patient goes in, they give a blood sample, the blood is then genetically modified in some way, and then that takes weeks, and then the modified cells get sent back to the patient. They get reinfused into the patient. That's autologous therapy. Allogeneic is the idea of an off-the-shelf treatment. You have some batch of cells, whether they're T cells or maybe they're derived from an iPS cell, or the others approaches have been using NK cells, but they're not derived from the patient. They're derived from robust, healthy donors. And you basically have like a bag of blood that's ready to go that could get infused into any patient. And that's another major way of reducing the cost. And also makes the product more uniform. It's not each patient having their own drug. There's an off-the-shelf drug that could be delivered to many patients. And then the third way that I'm actually very excited about and very optimistic about is in vivo therapies. This is like the extreme of the first way. If it's easier to make these cells like in smaller and smaller batches, and it could happen right at the patient's bedside, the extreme of this, which I think is really qualitatively different, would be could you just inject the vectors to do the DNA modification inside the body? So the cells never have to come out of the body, but a subset of all the immune cells would just get genetically modified inside the body, could make CAR T cells or other types. And we're seeing huge investments and breakthroughs in this using different technologies. There's companies now that are using viruses to deliver genetic instructions. Another company made a lot of progress delivering temporary instructions to T cells using lipid nanoparticles and mRNA. So I I think there's going to be a number of ways to potentially make these types of genetic instructions to tell cells inside of the body and really make these treatments much more accessible to broad numbers of people. I think first we have to figure out what the programs are that really cure disease. And I think we're now starting to see that that's not so far off, it's doable. And as we have those programs that work, I think the next step is how do we scale them up and get them to the patients that need it. And if you ask me, I think a lot of this is going to move in the in vivo editing direction or in vivo manipulation direction over the next couple of years.
Yeah, so I want to talk more about Arsenal. So I want to talk about like the journey behind it. Like, how did you get involved in, like founding Arsenal? Was it Sean Parker that gave you the idea to get into the whole like business entrepreneurship space, or was it someone else that was like, hey, maybe you should commercialize your research?
It was a mix of things. I certainly give Sean a lot of credit for it. I'll tell you a little bit. I'd actually been involved in setting up another company before that, which actually has since closed, which was a little bit heartbreaking. It was, I think, a company that was a little bit ahead of its time. It was an in vivo gene editing company called Spotlight. Um, but I had already learned a bunch about the idea of company creation from setting up Spotlight and had some ideas of what it would take to set up a company. And then as the technology really took off in our lab of what I just described, the ability to do non-viral insertion of big DNA sequences, I thought that could really revolutionize and have a big effect on how CAR T cells are made broadly. And the other piece of technology that was rapidly advancing in my lab was the ability to do massive scale genetic testing of many, many gene edits, being able to test essentially every gene in the genome to learn rules about which genes you might want to modify to enhance T cell therapies. So I thought there was a real potential for a company at this intersection of understanding what the genetic modifications were and then having a really powerful technology to make those genetic modifications in actual cell therapies. And I would say it was good fortune. Jeff Bluestone, who at the time was a friend and mentor to me at UCSF, was also the CEO of the Parker Institute. And Jeff I started showing him the work that we were doing, and he said, I think you should talk to Sean Parker about this. And I took him up on that. I think I kind of invited myself over to Sean's house. And Sean had me sit in his living room and we talked for hours about what is CRISPR, how does DNA editing work, what was the potential for cell therapy. We had a lot of far-reaching conversations about this. And we started floating the idea that maybe there was a company that could come out of this. I would say over the year that followed, Sean and I talked a lot, and we started amassing a team that brought together different expertise. And this turned out to be one of the really great things about starting Arsenal. We put together, I think, an all-star team of founders. I worked really closely with Nick Haining and John Weary, who are two of the world's experts on T cell exhaustion, epigenetic control of T cell state and function. Brad Bernstein, who's an expert in epigenetics and genomics, actually a former mentor and friend of mine from Boston. Uh, and then we brought in Theo Roth, who had done a lot of the work in my lab. And then after the company was founded, we actually added a really important element that had been missing. We brought in Cole Roybal as a founder, who has an expertise in synthetic biology and had been designing a new family of receptors that have special features for I could talk a little bit about it, but about actually connect to create a sort of two-factor authentication system where one receptor is designed to basically engage one target on a cancer cell, and only after that target is engaged, the first target's engaged, do you turn on a CAR T cell to engage a second target, which has potential to increase the specificity of what these CAR T cells will recognize and be able to improve the ability to discern between cancer cells and healthy cells. So we brought all these different pieces of technology and expertise together. We brought together, Sean really helped nucleate an amazing group of investors. And then we brought in an awesome CEO. Actually, one of the investors introduced Brooke Byers, introduced us to Ken Drazen, who's just been an absolutely visionary, determined, ultra knowledgeable CEO who I think has helped Arsenal navigate some pretty challenging waters of just the general time that's been quite challenging for biotech to stay funded and keep growing. So I give Ken a lot of credit for helping to refine what the actual products that Arsenal would design would be, and keeping the company moving forward to get to the core goals of what the company was founded for.
Yeah, so is Arsenal partnering with any like big pharma to manufacture its T cell therapy?
Arsenal does its own manufacturing.
Oh wow. So it's like expensive though.
Yeah. So I mean, in part because we have this specialized technology that came partly from my lab that's non-viral. Arsenal has put a lot of big investment in trying to make sure that the cells are manufactured in a way that's going to maximize the potency of these cells.
I see. Yeah, so I want to talk about CRISPR, like the whole ethical side of CRISPR. So I think like a couple years ago, like some Chinese scientist was like using CRISPR on some babies or something. So, what are some ways to make sure we can use CRISPR in a good way that doesn't create any concerns that scientists are creating babies?
Yeah. Well, you know, people can disagree on this, but I have a pretty hard line stance on this, which is that to me the ethical line is really on germline editing versus somatic cell editing. So basically, germline is a very small subset of the cells in the body, which basically are the cells that are either eggs or sperms or the cells that will go on to become egg or sperm. So those are the cells that carry the genetic information for future generations. I argue strongly that we should never intentionally edit the germline. That we just don't know what the future of genetic material we want. And the best thing we can do for future of genetics, in my strong opinion, is maintain natural diversity. That we want I think humans are really bad at staying away from fads. We tend to group around fads. And the last thing I'd want to see are fads of how we design our children in any way. Like, I think that there is a really strong need to maintain wild diversity in our human population for all sorts of reasons to enhance the quality of our society and just make sure that we have a broad, diverse gene pool for future generations. So I am strongly opposed to any kind of germline engineering. That said, I think, we have enormous power to potentially make a difference in patients' lives. I don't want to ignore that. And I think we should be aggressively pursuing using CRISPR in somatic cells, making genetic edits in cells that are where there's mutations or disease that are causing disease, or where potentially in T cells where we have an opportunity to override disease to treat the underlying cause of disease. That won't, we make genetic edits that don't get passed on to future generations. And to me, that's essentially like using these genetically engineered cells like a drug. You talked about this idea of living drugs. I think that phrase actually comes from Michelle Satterline in New York. This idea that if you make a genetic edit to the cell, you have some cell that can go around and have a period where it's acting as a drug. It can recognize cells that it wants to eliminate. It can act in ways that we've programmed. But like a drug, I think these cells have a half-life. So it can be quite long. Some of these cells persist for years, maybe decades, but it won't go on to the next generation the way it would with germline editing. And so to me, that's a line that makes sense ethically, and I've been quite outspoken in pursuing that.
Yeah, so I know 2017 you wrote like a lost article asking the US government not to cut funding
No,
for gene research. So now I know Trump is cutting a lot of research. So how are you responding to these cuts as a PI? Like, are you looking through your balance sheet to like make sure you're cutting any projects that could could be using up some of these funds?
it's a challenging time for science in America. It's an amazing time for science in America too. I do want to emphasize that. We have never had more tools to understand human biology, and we've never had more tools to intervene to treat patients in different ways. So we can sequence genome, we can use CRISPR to test every gene in the genome. We can leverage all the data that we have with AI, and the ability to get insights that would otherwise have been unimaginable. We're living in the future. like this is all happening in science. There's also simultaneously a crisis of faith in science. I think there's a broad skepticism of science that is alarming and exacerbated by a number of things that happened, I guess, exacerbated by things that happened during the pandemic. And for a number of reasons, science funding is under serious threat. I don't know, we don't know the scale of how serious that threat is, but the administration's clearly made some moves to try to cut funding substantially. My strong view is that since the end of World War II, there's been an incredibly successful partnership between the US government and research institutions, academic research institutions, private research institutions, to advance fundamental knowledge and to create the engine for a whole biomedical sector, for a whole innovation sector that has been a bedrock of the American economy and provided innumerable drugs. This is something that was laid out by Vannevar Bush at the end of World War II at the request of Franklin Roosevelt and delivered to President Truman. And there was a clear vision of the government should partner um, with research institutions and academic institutions and independent research institutions to train the next generation of scientists to create fundamental knowledge that might be missed in industry that would fuel subsequent industry. And it's happened. It's all happened, and it's advanced human health and advanced the American economy. And I guess it's being renegotiated now. It's been a partnership that's been successful that has been renewed by administration since 1945 essentially, and it's being renegotiated now. I think a lot hangs in that balance of this has been a partnership that has paid enormous dividends, and I hope it continues. So I guess that's my take on where we are, and I can't overstate the importance of how that gets negotiated now.
Do you think like biotech companies or like private foundations could kind of take over that funding a bit more a bit in the future if the government starts cutting more?
I guess I don't want to give, it's a complicated answer. I think what we're seeing now is that, I have seen amazing resilience where there's been some effort to step up and various groups, philanthropy, foundations, maybe to some extent industry have tried to fill this gap, but there's no sustainable way to continue this without the support that's come from the federal government. We're talking something on the order of $40 billion a year, and there's no other source for that money. So if we want science to continue to look the way it has, and I think sometimes this gets a bad reputation, it gets parodied in the public perception, but you know, investing in science that we don't yet know what's going to be profitable has been the role of the federal government, right? I mean, I think it's very easy for industry to come in and say, "Oh, look, something is teed up. This we see how this could be a drug in 5 years or 10 years." But if you're talking about basic mechanisms of biology, the question is not how will this be a drug, but how do we understand something that we just don't understand? Industry doesn't step in and fund that. It's very hard even to get most philanthropists to see the importance of that. There's some incredibly forward-looking philanthropists who are just excited about the potential of science, but that early investment in science before there's a clear profit, before there's a clear positive present value, that's where where those government dollars have had really outsized influence on maintaining the ongoing potential of American science.
Yeah. So, what advice do you receive during your career that shaped your professional development or success?
Um, I'm hesitating not because I didn't haven't got it, but because I've gotten so much. Um, that I think I've heard in various formulations, and if this could be probably be attributed to different people, but I think one of the things that is the most important in science is hard. The basic goal of science, of scientific training, scientific work, is to do something that no one else has ever thought of, or that people have thought of, but that has been impossible. And those are pretty tall order. And I guess the advice that sticks with me the most is if you're going to do that, make sure that you're enjoying it, that you're passionate about it, but also that you're not wasting time on things that are not the most important. That there's so much work that goes into any scientific project. You can delude yourself into thinking you're doing something easy or taking a shortcut, but really you need to be guided about where that effort that can be and will be enormous if you're going to be successful in any form of science, that you should be directing it at something that's the most important. And that doesn't have to be the most important short-term thing for patients. There's a whole spectrum of where science can have importance. But knowing what's guiding you and being really intentional about picking the project where you can make the largest contribution or where you see a huge opportunity rather than just going forward with emotions because you're going to still put in a lot of work with science.
Yeah, thank you, Dr. Marson, for being part of this episode so people can know more about your insights into T-cell therapies.
Thank you so much for having me, and thanks for providing a forum to talk about science, which remains one of the most exciting things that we can do.